So this is a picture from your book. You can see that it is a pretty complex enzyme. In fact, it is so big that you can visualize it under an electron microscope. It consists of two large subunits: the F0, which is inside the plasma, which is inside the inner mitochondrial membrane, and its face is the inner mitochondrial space. And this F1 subunit, which is actually the catalytic subunit, and it catalyzes the conversion of ADP to ATP, called F1, which is protruding into the matrix of the mitochondria. And this is what you see here. This is the little C ring, and this is the much less focused. I'm just trying to make, the reason I'm being quiet is I'm trying to make sure I'm correct. I doubt my statement, guys. I think this picture is inverted. I think this is the alpha and beta subunits of the F1. This is the hook of the delta subunit right here, and the glow right here is from the actually non-existing C ring, because they wouldn't have been able to put it just on a matrix of electron microscope because it has to be solved in lipids. So instead, this whole thing was attached to some sort of glowy bead. So, and another bit of ATP synthase trivia is that they don't actually stick out of the membrane like that. They dimerize, and then these little dimers form superstructures, which actually cause the involution of the membranes and cristae, because they solidify the membrane and force it into that particular cristae shape. And here is a video to show you the fantastic little ATP synthase in action. Now, this is a model. This is not the actual ATP synthase somehow being filmed, but it is a model based on crystal structures of all the ATP synthase complexes. And we will talk about the proof that this ATP synthase actually spins in the next few slides. And the top link right here is a link to this movie that you're going to see. And the bottom link is an alternative movie where there's actual narration that you can, which you can go through on your own terms. So here we have our ATP synthase. This is the membrane. This is a rotating C ring. And these, you can see that these subunits move as the ring rotates. This is the F1 portion with the red and yellow alpha and beta subunits. And this blue guy here is the rotor that makes them move around. You can see how when the rotor moves, and the rotor is being moved by the proton motive force, it bumps the subunits and introduce conformational changes. So let's move on. We'll go back to that. So you guys saw the alpha and beta subunits of the F1 complex move around in the video, and the beta subunits actually go through 3 different conformation stages. A conformation called open form, when ADP and phosphate can enter the active site and be held in place by side chains of the amino acids of the active site. The loose conformation, where the nucleates are now trapped inside the subunit. And then the tight conformation, where they actually, the phosphate and the ADP are literally shoved against each other, allowing for the bond between the third phosphate and the ADP molecule to form, forming ATP. Now, the important thing to realize here is that ATP synthase can actually run backwards. If for some reason the proton motive force, if the matrix is more acidic, then when there's more protons in the matrix than there are in the inner membrane space, ATP synthase, the protons will flow the other direction through the C ring, and that will result in their the little turbine rotating in a different direction, and it will actually start binding ATP and then breaking it apart. So the way the ATP synthase rotates is very important for it to be able to perform catalysis, because the ADP and phosphate have to bind first, then in the loose form they're trapped in the beta subunit, and then in the tight form the phosphates can be shoved against each other, causing just holding the phosphate and the ADP molecule in place long enough for the third covalent, fairly unstable bond to be formed. And this is a summary from your book how this is happening. You can see how there's a rotation occurring here. The ATP is released in the open, so how the open side becomes the loose side, then becomes the tight side, and so forth. And this is the graphic, a GIF image I got from Wikipedia, really, which also shows the same idea. Now, in this image here, there's still white subunits, which are the alpha subunits of the F1 complex. They don't have any catalysis, they're just there to take up space. In this image, the alpha subunits have been completely dropped, and instead you can see the rotation of the three subunits. Just pretend the alpha subunits are the bicolored bits. So you can see here that in this image, you have the open conformation where molecules can come in and out. ATP can leave, ADP and phosphate can come in. This is the tight conformation, where now the ADP and phosphate are held together, are held by the active site and can't leave. And this is the tight conformation of the enzyme, where the ADP has been shoved against the third phosphate and has formed ATP. Let's see if it's going to work. There we go. Leaves, enters, leaves, enters. So you can see, depending on the three conformational shapes of each of the beta subunit, they will perform a different type of reaction, a different type of process. Release, rebind, catalyze. So let's look at that same process now in a cool video that I found. Alright, there we go. So, one important thing about this particular video is because this video was made true to the crystallographic structure that they were able to obtain. So in this case, the ATP synthase is actually rotating backwards. It's taking up ATP and breaking into ADP and phosphate, because this is a far easier situation to set up in the lab for crystallizing all these different conformations, because it's hard to crystallize a process. A crystal of a protein is a static thing. So crystal structures like that are obtained by getting a bunch of little snapshots, by making the proteins take in a bunch of different conformations. There we go. So now it's gonna break it apart. There we go. It broke it. Even though in our cells it actually takes ADP and phosphate and makes ATP. We're just running in reverse here. So you can see in this particular, these are the side chains. By gosh, you don't need to know the side chains. This is ADP just came in, and you can see what happens. These two guys are holding, kind of tugging on the phosphates, and in this case breaking it away from the ATP molecule. In a real-life situation inside our mitochondria, instead, the phosphate and the ADP would be pushed against each other, causing for this bond to be catalyzed. There we go. Another subunit came in and pushed it, and pushed the phosphate away from the molecule. Now, in the real situation, we'd be bringing it back in. So, all of this, as I said, crystal structures are static, so they don't actually visualize the motion of the motor. However, just in the previous slide, I said that we know that the ATP synthase performs like 1.3 rotations per second. Well, how do we know that? So what has been done is these just the F1 subunit of the ATP synthase was attached to a surface, in this case a glass slide, using various tricks. And the gamma subunit has an actin filament attached, something that's very easy for us to visualize because we can show glowy stuff in between these 8 actin subunits. And actin, of course, is part of our, it's a structural protein which is part of our cytoskeleton. So let's watch this particular video of ATP synthase, the gamma subunit actually rotating, and thus you can watch the actin filament, which is much bigger than the gamma subunit, also rotate. And it does so fairly fast. So here, this guy here is the glowing actin filament, because you can't visualize the ATP synthase itself. It doesn't glow. And if you start attaching stuff to it, it doesn't work. So here we go. There's the guy. Do you see how it goes? Here it goes. Rotating away, working hard, and totally acting like a little motor. Now, there's a wealth of information about ATP synthesis on YouTube, though I would like to warn you about the various creationist videos that are out there as well, because the majesty of ATP synthase has made some people think that it couldn't have been possibly created by nature. It must have been designed by a supernatural being of your choice. And there's a lot of arguing going on in the creationist community as to, how the science is lying to us. But in fact, there's multiple labs working on trying to figure out exactly how ATP synthase evolved, what it originated from, and how these complicated protein machines came to be to begin with. And there's actually strong parallels between. Now, this is a work in progress. There's no actual answer here, same as with the lost city alkaline vents. But there's a lot of connections between the sequence of ATP synthesis and other proteins. One of them is the bacterial flagellum, but others, even more rudimentary proteins, are known as helicases. They unwind DNA, and also protein motors, protein pumps that use the rotational motion of the channel in order to move proteins from one membrane compartment into another. Now, you can see that the bacterial flagellum is also has a very much ATP synthase-like unit here. We have our little C here. We have the purple guys is what looks like the C ring that pumps protons, and the motion of the C ring and the pumping of protons is coupled to the rotational motion of this massive, massive protein structure of the bacterial flagellum that allows them to move. And some bacteria have 1 flagellum, some have 3, and some have a bajillion. And aside from using proton motive force to spin their long flagellum, bacteria have other things coupled to proton motive force power, such as excretion of proteins.